Hello everyone and welcome to this CoBCoM lecture. I am Sarah Sedlar, a PhD student in Athena project team at INRIA and at University Côte d'Azur. I will present you a work done together with Abib Alimi, Theodore Papadopoulou, Rashid Erish and Samuel Delorier-Gauthier on spherical convolutional neural networks for diffusion MRI analysis. We have two publications related to this work: Spherical UNet for Fiber Orientation Distribution Function Estimation, presented at dMRI workshop in 2020, and Fourier Domain Spherical CNN for Microstructure Parameter Estimation, which is accepted to MICAI Conference 2021. I will start with a brief introduction to diffusion MRI local modeling, in particular to modeling via multi-compartment models and fiber orientation distribution functions. Further, some general challenges related to the analysis of discrete spherical signals are provided. After that, I will present you our work on spherical CNN models designed to address two different problems in diffusion MRI local modeling. So, diffusion MRI is an in vivo and non-invasive imaging modality which uses diffusion sensitizing gradients to create contrast. These gradients are characterized by multiple parameters such as direction, strength, shape, and duration, which all together constitute a high-dimensional acquisition space, also termed as Q space. Acquisition of diffusion MRI signals over multiple points of the Q space can provide information on how diffusing water molecules interact with surrounding tissue structures, and as such it is well-suited imaging modality for white matter microstructure imaging. Top left figure contains an illustration of a neuron, and the right one hydrogen protons of diffusing water molecules which are trapped in an axon. Diffusion MRI signals measured along the gradient direction, which is perpendicular to the axon, will have high value since diffusion of the molecules along that direction is restricted. On the other side, a signal measured along axon will have low value due to non-restricted molecule diffusion. A single voxel contains thousands of axons, which constitute axon bundles, and some of the possible axon bundle configurations within a single voxel are illustrated in the 5 bottom figures on the left. Keeping all gradient parameters constant except their direction corresponds to diffusion MRI sampling on sphere, which is also called shell. Single shell sampling scheme is depicted in the left figure, and if in addition to direction gradient strength is changed as well, this will correspond to multi-shell diffusion MRI sampling as illustrated in the right figure. In this slide, you can see illustrations of diffusion MRI signals acquired over 3 shells for one axial slice of a brain. The most important properties of these signals are that they are real and positive, antipodally symmetric, signals associated to individual fibers are axially symmetric, and diffusion MRI signals are corrupted by non-additive Rishan noise. One way to model measure diffusion MRI signals is via fiber orientation distribution functions, or shortly fODFs. An fODF is a spherical probability density function that reveals orientation of underlying axon bundles and their volume fractions. Figure on the left illustrates signals in 2 voxels acquired over 3 shells, one voxel containing single and another one crossing axon bundles. Corresponding fODFs are depicted in the figure on the right. Estimation of the fODFs over entire white matter region is a necessary step for tractography, which is crucial for the analysis of structural brain connectivity. Starting from the fODFs depicted on the left, using tractography algorithms, one can obtain tractogram as depicted on the right. Multi-compartment microstructure imaging is another family of approaches for local diffusion MRI modeling. These models are biophysically inspired and represent measured signal as a linear combination of signals coming from different tissue compartments, such as intra- and extra-axonal spaces, gray matter, cerebrospinal fluid, and tumorous cells. Several studies have shown that parameters associated to some of these models have potential in the evaluation of several neurological diseases and in the characterization of the early brain development. One prominent multi-compartment model is the Neurite Orientation and Dispersion Imaging Model, or shortly NODI. It represents a diffusion MRI signal as a linear combination of three compartments, namely intracellular compartment for modeling of axons and dendrites with cylinders of zero radius distributed according to Watson distribution, then extracellular compartment for glial cells and neuron cell bodies, modeled with anisotropic Gaussian with orientations that also follow Watson distribution, and the last compartment is for cerebrospinal fluid that is modeled with simple isotropic Gaussian. In the following few slides, I will present you some challenges related to the analysis of discrete spherical signals. Euclidean signals are most commonly discretized on equidistant sampling grids, as illustrated in the top figures. In analogy to Nyquist-Shannon sampling theorem for Euclidean signals, a number of theorems on sphere have been defined as well. These sampling grids guarantee that all the information from a band-limited continuous spherical signal is preserved after discretization. An example of such a sampling grid is provided in the bottom left figure. However, since in reality the signals are often corrupted by significant noise and are not band-limited, equiangular sampling grids with excessive sampling around poles are often not optimal. In diffusion MRI, in the last decades, among the most commonly used sampling schemes is multi-shell Q-sampling, where points are uniformly randomly distributed over single or multiple shells, as illustrated in the bottom figure on the right. This sampling scheme provides the most optimal angular coverage in diffusion MRI imaging. To analyze discrete signals, we are often using convolutional filters with certain properties. In the case of Euclidean signals, convolutions between two, for example, 2D signals gives a signal in 2D as well, and in general there is a correspondence between signals and kernels sampling grids as illustrated on the left. On the other side, convolution between spherical signals gives a signal in SO3 manifold, and in addition there is no correspondence between signals and kernels sampling grids as illustrated in the figure on the right. This means that convolution in signal domain would require interpolations, which can be very computationally expensive. As it is well known, integrable Euclidean signals can be represented as a linear combination of elements of Fourier basis via Fourier transform, as illustrated in this slide. In analogy, an integrable spherical signal can be represented as a linear combination of spherical harmonics. An example of diffusion MRI signal is given in the left figure. Elements of spherical harmonic bases of different orders and different degrees are depicted in the middle, and associated spectral coefficients are on the right. Since with any type of spherical sampling, orthogonality of spherical harmonic bases is lost, inversion cannot be performed by simple transpose. Each equiangular sampling theorem has associated quadrature weights for computation of spherical harmonic coefficients. And other approaches which can be used with arbitrary sampling grids include the Gram-Schmidt orthonormalization process and least mean square solutions without or with regularization terms, such as Tikhonov and Laplace-Beltrami term, which is well suited to spherical harmonics. In this work, we have investigated how to adjust CNN models to the properties of diffusion MRI data and how to use these models to estimate local functions and parameters from data acquired with clinically applicable acquisition schemes. In the figure on the left, an example of sampling scheme from Human Connectome Project containing 270 points over 3 shells is illustrated. Acquisition of one diffusion MRI scan with this large number of sampling points lasts around an hour, which is not clinically desirable. In the figure on the right, an example of a reduced sampling scheme used in our experiments is illustrated, and it contains 60 points per 3 shells. In the following slides, I will present you our work on spherical U-net for fiber orientation distribution function estimation. I will start by presenting main building blocks of a U-net, namely convolutional pooling and transposed convolutional layers. Among the first prominent spherical CNN models was the spherical CNN proposed by Cohen et al. The main novelty in their work are convolutional layers, where convolutions between signals and kernels are performed in spectral domain in order to avoid computationally expensive interpolations which are required in signal space. The model takes as input S2 signals sampled at equiangular grids, which are transformed to spectral domain using corresponding quadrature formula. In the first convolutional layer, convolution between signals and kernels is performed as a simple outer product of corresponding spectral coefficients, as illustrated in the figure on the left. Resulting coefficients correspond to spectral coefficients of SO3 feature maps. Apart from the first convolutional layer, all the following take as input SO3 feature maps and perform convolutions with SO3 kernels, again in spectral domain, by simple product between corresponding spectral coefficients, as illustrated in the figure on the right. To apply rectified linear unit nonlinearity, signals are transformed from SO3 spectral to signal domain, where the nonlinearity is applied in accordance to planar CNNs. Since transformations between spectral and SO3 signal domain only to apply nonlinearity are still computationally expensive, Esteves et al. Proposed a model which contains axially symmetric zonal kernels. Convolution between an S2 signal and a zonal kernel gives a signal in S2 as well. As in the work of Cohen et al., model takes as input S2 signals sampled at equiangular grids. Spectral coefficients are computed using quadrature formula, convolutions are performed as product in spectral domain, and nonlinearity ReLU is applied in signal space. Another layer type used in standard CNNs and in UNets is pooling layer. Task of a pooling layer is to summarize a feature map by taking mean or max over a small neighborhood and to provide feature map of lower resolution to the following layer. In both works of Cohen and Esteves, this was achieved by discarding high-frequency spectral components, as illustrated in bottom figures. Transposed convolutional layer is a type of layer essential in the architectures of UNets. It learns estimation of feature maps of higher resolution or extrapolations. In analogy to planar UNets, transposed convolution is performed by insertion of zero samples into input feature map, followed by convolution, which is in the case of spherical UNet, again performed in spectral domain, as illustrated in the bottom figure. Putting together convolutional pooling and transposed convolutional layers, we have designed a spherical UNet for fiber orientation distribution function estimation. As input, it takes multi-shell diffusion MRI data sampled at uniformly randomly distributed points from a single voxel or a small 3D neighborhood, as illustrated on the left. Spherical harmonic coefficients are estimated using Gram-Schmidt orthonormalization process. Since diffusion MRI signals are antipodally symmetric and signals associated to individual axon fibers exhibit axial symmetry, convolutional and transposed convolutional layers contain zonal antipodally symmetric kernels. As planar UNets, it is composed of a contracting and an expanding part. Contracting part contains convolutional and pooling layers which extract relevant features of different resolutions. Pooling is achieved by low-pass filtering simply by discarding high-frequency components. Expanding part contains transposed convolutional layers which combine feature maps from expanding and from contracting part in order to perform higher resolution extrapolations. In both convolutional and transposed convolutional layers, ReLU nonlinearity in signal domain is used. In the experiments, we have used real data from Human Connectome Project and synthetic data generated using dMRI toolbox following HCP acquisition protocol containing 270 points over 3 shells. Gold standard is estimated using a state-of-the-art method, multi-shell, multi-tissue constrained spherical deconvolution, applied on diffusion MRI data acquired on full sampling scheme. Methods are evaluated and compared on diffusion MRI data acquired over a reduced number of sampling points, varying between 20 and 120 points over 3 shells. We have compared multi-shell, multi-tissue constrained spherical deconvolution, 3D convolutional neural networks with 3D neighborhood as input, and our spherical U-net with different number of trainable parameters and different sizes of inputs. We have used in total 50 HCP subjects, 30 for training, 10 for validation, and 10 for testing. In this slide, comparison of the methods in terms of mean squared error for different number of sampling points is provided for real data in the figure on the left and for synthetic data in the figure on the right. We can notice that for lower number of sampling points, using neighborhood information is essential, which explains large accuracy gap between multi-shell, multi-tissue CSD and spherical U-net with single voxel input, compared to deep learning approaches that take into account neighborhood information. Further, we can notice that our model with neighborhood input provides improvement over 3D CNN, even with the model with smaller number of trainable parameters. Higher differences on synthetic data indicate that our model is more robust to noise. In this slide, you can see differences between mean squared errors for fODFs estimated from 60 sampling points. Differences are illustrated for white matter region of one axial slice between multi-shell, multi-tissue CSD and our approach on the left, and 3D CNN and our approach on the right. We can notice that our approach yields lower mean square error in large majority of the voxels, and that these differences are more important with respect to the multi-shell, multi-tissue CSD. We have also compared models in terms of mean angular errors over voxels containing single fibers in the figures on the left and over the voxels which contain crossing fibers in the figures on the right. We can notice that for single fiber bundles, for real data, performances of our model and multi-shell, multi-tissue are almost equal, while on synthetic data, our approach provides some improvements. More significant differences are present in the voxels containing crossing axon bundles, especially for lower number of sampling points, again indicating importance of using neighborhood information as input. In this slide, you can see qualitative comparison of fODF estimations with underlying ground truth fODF peaks. Here we can notice that multi-shell, multi-tissue CSD is more prone to produce false peaks, and on the other side, deep learning approaches sometimes tend to provide over-smoothed fODFs. This smoothing is more prominent in the case of 3D CNN. At the end, I will present you our recent work on Fourier domain spherical CNN for microstructure parameter estimation. Since nonlinearities applied in signal domains such as ReLU might introduce aliasing, a fully Fourier domain spherical CNN termed as Clebsch-Gordan network has been proposed by Condor et al. Instead of SO3 signals, activations of each layer are SO3 covariant vectors or fragments. After convolution, which can be seen as a convolution with zonal kernels, nonlinearity of quadratic nature is applied using Clebsch-Gordan decomposition of tensor product of input fragments. From each layer, SO3 invariant scalars are extracted, which correspond to fragments of degree 0, and they are fed into fully connected network. Since in Clebsch-Gordan network increase of channels at the output of each layer is quadratic, we have investigated a model that preserves SO3 activations and uses channel-wise SO3 quadratic nonlinearity. In addition, from each layer we have extracted power spectrum features which are also rotationally invariant. Architecture of our Fourier domain spherical CNN is illustrated in this slide. The model takes as input diffusion MRI signals acquired over multiple shells. It contains denoising layers, which are followed by spectral coefficient estimation using Gram-Schmidt orthonormalization process. Convolutions are performed in spectral domain, and as in the work of Cohen, the first layer contains convolutions between spherical signals and kernels, and the following ones between SO3 feature maps and kernels. After each convolutional layer, quadratic nonlinearity is applied and pooling by discarding high-frequency components. From the inputs to each convolutional layer, we have extracted rotation invariant power spectrum features, which are fed into a fully connected network. Since diffusion MRI signals are affected by non-additive Riemann noise and are by nature positive, we have defined denoising layers as mapping performed with matrices that are close to identity, followed by ReLU nonlinearity, which guarantees that the resulting signals are not negative. S2 and SO3 convolutions are performed in spectral domain as in the work of Cohen et al., but contrary to their model, kernels are represented and trained in spectral domain as well. We have defined channel-wise quadratic nonlinearity, which is applied on SO3 feature maps in spectral domain. We observed that quadratic function of SO3 signal f can be represented as a linear combination of elements of Wigner-D matrices, which can be seen as elements of Fourier basis for SO3 signals. We also observed that product in signal domain of SO3 signals in spectral domain corresponds to Clebsch-Gordan decomposition of tensor product of corresponding spectral coefficients. Using generalization of Parseval's theorem, rotation invariant power spectrum features of the input S2 diffusion MRI signals and of the extracted SO3 feature maps are computed for each spectral degree L. They are concatenated into a rotation invariant feature vector, which is fed into a fully connected network. We have used in the experiments real data from Human Connectome Project, and the methods are evaluated and compared on the problem of no-D parameter estimation, namely intra-axonal volume fraction, isotropic volume fraction, and orientation dispersion indices. Gold standard no-D parameters were estimated using brute-to-fine optimizer from the MyPy library applied on diffusion MRI data acquired with full HCP sampling scheme containing 270 points distributed over 3 shelves. Methods are compared on clinically desirable reduced sampling scheme containing 60 points over 2 shelves. We have compared our method to brute-to-fine optimizer and 2 deep learning approaches, namely fully connected network and microstructure estimation using a deep network, or shortly MEDN. For training of deep learning approaches, we have used 30 subjects. Validation subset contains 10 subjects, and final testing was performed on 10 subjects. Methods are quantitatively compared in terms of mean absolute errors, averaged over 10 testing subjects, and obtained results can be seen in the table. We can notice that deep learning approaches significantly outperform brute-to-fine optimizer. Comparing our methods to another 2 deep learning approaches, we can see that it yields lower average error for all of the 3 compared no-D parameters. To investigate rotation invariance of deep learning approaches, we have trained models on the data whose diffusion tensor feed direction is in the range between 0 and 30 degrees. The quantitative results of the experiments are presented in the table, and they clearly indicate rotation invariance of our model, with the most important difference in the estimation of the orientation dispersion indices. In this slide, you can see qualitative comparison of no-D parameter estimation and differences with respect to gold standard, where red color indicates underestimation and the blue color overestimation. Whereas the differences between brute-to-fine and deep learning approaches are noticeable, some differences between deep learning approaches can be seen in OD estimation, where fully connected network and MEDN tend to overestimate orientation dispersion indices compared to Fourier spherical CNN. In this work, we have investigated how neural network models can be adjusted to the properties of diffusion MRI data and how they can be applied to the problems of diffusion MRI local modeling. We have shown that spherical CNNs exhibit higher generalization power and can provide some improvements compared to the models which do not take into account nature of diffusion MRI signals. In the future work, we will investigate another spectral domain nonlinearities, how neighborhood information can be integrated into Fourier domain spherical CNNs, and also we will investigate some another deep learning architectures such as Fourier domain spherical units. We are grateful to INRIA Sofia Antipolis Mediterranee, NEF Computation Cluster, and to the Opal infrastructure from Université Côte d'Azur for providing resources and support. Part of this work has been supported by the French government through the Troisième Côte d'Azur investments in the future project managed by the National Research Agency. This work was funded by the European Research Council ERC under European Union's Horizon 2020 Research and Innovation Programme, ERC Advanced Grant Agreement number 694665 PI Rashid Arish, CoBCoM Computational Brain Connectivity Mapping. Thank you for your attention.